Ray Nims is another of our speakers who has shared his wealth of knowledge with us at previous annual meetings. Since 2009, Ray has served as a consultant subject matter expert to the pharmaceutical industry in the areas of viral testing, viral safety, animal-derived material risk assessment, and quality. Previously, Ray was a subject matter expert at Amgen, directing viral and mycoplasma testing of raw materials and products, and managing Amgen's global contract analytical testing lab outsourcing. From 1994 to 2006, Ray was employed by BioAlliance with many similar responsibilities. He currently serves on the editorial board of the Bioprocessing Journal and has served on advisory boards for several USP chapters. He is a widely published generalist in the biomedical sciences. Today, he will share his perspective on the validation of serum gamma irradiation, and in particular, who should be making the decisions to irradiate or not. So I'm very pleased to introduce you once again to Dr. Raymond Nims.
Hi, Rosie. Well, here we are. It's 2021. We still can't have a face-to-face meeting. It's too bad. Well, we'll make the best of it. This year, you asked me to talk again about gamma irradiation, and this year's title is Validation of Gamma Irradiation. Who should be driving the bus? Let me get my slides up here. Here we go. And I addressed this question in two ways. One is I look back at the past literature to see who published in the past on gamma irradiation validation, specifically for serum. And secondly, I looked at the serum guidance to see if that might shed any light on responsibility around validation. So before I start, I will remind you that gamma irradiation of serum is a barrier treatment intended to mitigate, but not necessarily eliminate, the risk of introducing a viral contaminant into a cell culture. Known contaminants that have been attributed to serum use in the past include these viruses: reovirus, blue tongue virus, epizootic hemorrhagic disease virus, Cache Valley virus, and of course, bovine viral diarrhea virus. In addition, a couple of viruses have been suspected to be attributed to the use of serum. These include vesivirus 2117 and bovine polyoma virus. But in addition to risk of introducing viruses into cell cultures, non-barrier treated serum obviously represents a vehicle for importation of non-indigenous viruses into a region. In non-gamma irradiated bovine serum, the risk of viral contamination is mitigated only by sourcing considerations such as geography, immunization status, oversight of abattoirs, and testing. And testing, is not a very robust risk mitigator. Why? Because serum lots are large, maybe 3,000 bottles, and typically one bottle would be sent for testing. Hence, the need for barrier treatment. So why validate the irradiation process? Well, validation is what tells us how effective the barrier treatment is for reducing possible infectious virus load in the serum. We do this by testing for viral inactivation efficacy following gamma irradiation at the planned kilogray dose range and under the specific irradiation conditions to be used by the irradiator. So you might ask, why can't we just use a higher irradiation dose and kill everything? The problem is that cell growth performance of the serum is also impacted at higher irradiation doses. And such performance effects are really the limiting factor for using irradiation. Now, to get to the crux of the question for today's topic, who should validate the irradiation process? Historically, gamma irradiation validation has been conducted by 4 types of entities: serum suppliers, who sell irradiated serum and make claims as to efficacy; regulated serum users, making claims as to barrier treatment efficacy; serum users, and these are all users, not only regulated. They should ensure that the irradiated serum will perform in their specific application as well as the non-irradiated serum. This is really not a validation, but a qualification. And finally, regulatory agencies have participated in the past in characterizing the efficacy and the impacts on cell growth following gamma irradiation as a barrier treatment. What exactly is meant by validation of gamma irradiation? And this really depends on the entity conducting the validation. So, for instance, for serum suppliers, validation typically means evaluating viral inactivation efficacy, as well as impact of the irradiation on serum performance. In the case of serum suppliers, validation means establishing the irradiation process from start to finish, where start is shipping of the serum to the irradiator, and finish is shipping the irradiated serum back to the supplier. And of course, the dose range of the irradiation to be routinely used is established. USP 1024 is a chapter called Bovine Serum. This section has been taken directly from USP 1024. It says that validation of gamma irradiation has 2 aspects. 1, dose delivery in a defined load configuration, and 2, an activation capacity. Critical irradiation process parameters include product serum temperature, packaging size and configuration, dosimeter distribution, and defined minimum and maximum dose received. Dosimeters should be used to monitor the established high dose and low dose positions in each irradiation run. And here's the important clause right here. If the serum manufacturer makes an activation claims, these should be supported by the manufacturer's own well-designed viral inactivation studies. To a regulated user of an irradiated serum, validation typically focuses on inactivation efficacy for specific viruses of concern to the manufacturing process, and again, demonstrating lack of adverse impacts on product quality that might have occurred using irradiated serum compared to the use of non-irradiated serum. Again, for a non-regulated user of an irradiated serum, validation is not really done. It's more of a qualification, just focusing on demonstrating equivalent serum performance in the application compared to use of non-irradiated serum. To a regulatory agency, validation typically focuses on demonstrating efficacy for inactivating viruses of importation or biologics manufacturing concern, while at the same time maintaining satisfactory serum performance compared again to the use of non-irradiated serum. In this section, I wanted to give you the actual citations coming from the literature. I won't go through this all. And I should mention that the citations are provided at the end of the talk in the slide deck in case you're interested. So this section is on serum suppliers who have validated the irradiation of serum. We've got representation from Hyclone, Life Technologies, SAFC Biosciences, and JRH Biosciences. And you can see that these citations have included both inactivation efficacy and impact on cell growth. Just a few citations from regulated serum users making claims as to barrier treatment efficacy. This included Amgen evaluating the irradiation of serum to prevent introduction of Cache Valley virus, reovirus type 3, SV40 as a bovine polyoma virus surrogate, and mycoplasma in FBS. And I found this from Razi Vaccine and Serum, evaluating the efficacy of gamma irradiation of calf serum this time, using bovine viral diarrhea virus and infectious bovine rhinotracheitis virus as challenges. And this paper also discusses impacts on cell growth. All serum users obviously should make sure that irradiated serum will perform in their specific application prior to switching from non-irradiated serum. Unfortunately, these types of studies are not typically reported in the literature. And then as far as regulatory agencies, I did find a couple of articles. USDA back in 1990 looked at efficacy for inactivating viruses of importation concern. And these included Acobane virus, Inovirus, bovine ephemeral fever virus, swine vesicular disease virus, foot and mouth disease virus, hog cholera virus, blue tongue virus, and minute virus of mice. Again, this is in fetal bovine serum, frozen. And Erickson et al. Examined serum performance following irradiation. Let's look at some of the bovine viruses of concern since we're talking about these. And this list of viruses comes from 9 CFR 11347. It's law. The title is Detection of Extraneous Viruses by the Fluorescent Antibody Technique. And it lists the viruses of concern for different raw materials of animal origin. So these are the bovine viruses of concern. Bovine viral diarrhea virus, reovirus, rabies virus, blue tongue virus, bovine adenoviruses, bovine parvovirus, and bovine respiratory syncytial virus. Interestingly enough, this list has a title, Requirements for Ingredients of Animal Origin for Production of Biologics. Although this title suggests that the list might have been formulated by FDA, it actually reflects the concern of the Animal and Plant Health Inspection Service, APHIS, of the USDA. And, this is a fairly old list, and rightfully so, the USDA is currently looking to update this list of viruses to reflect newer emerging viruses. The World Health Organization has specified a few additional viruses to add to this 9 CFR list. These include infectious bovine rhinotracheitis virus, parainfluenza type 3 virus, and vesicular stomatitis virus. There's your citation for that. The viruses of concern from the biologics manufacturing point of view, again, these are viruses that have been attributed to the use of serum in manufacturing processes. Epizootic hemorrhagic disease virus, which is not a 9 CFR virus of concern. A reovirus type 2, blue tongue virus, yes, these are 9 CFR viruses. And then these last two, vesivirus 2117 and Cache Valley virus, are not on the 9 CFR list. Biologics regulatory guidance tells us to test for these 9 CFR viruses, but also to be aware of emerging bovine viruses and to test for those where appropriate. So again, this is coming from EMA guidance. Some of the more current viruses of importation concern are listed in this slide. We've got the old 9 CFR 113 viruses plus bovine rhinotracheitis virus, and then some new ones: bovine coronavirus, bovine rotavirus, bovine leukosis virus, and Schmallenberg virus. I'm going to talk about these in a little bit more detail. Bovine coronavirus We don't have a lot of detail. We don't have much information about coronavirus inactivation as a family in frozen serum. What we do have, though, is some data on frozen culture medium containing 2.2 to 5% FBS. And that's listed in this table. You can see SARS-CoV-2 and SARS-CoV and MERS-CoV. So we've got this data. It's done at minus 80 degrees, which is appropriate. And then it's in culture medium containing small amounts of FBS. But importantly, the efficacy, which is shown here, log 10 inactivation per kilogram, it's very high. So I mean, what this means is if 0.92 logs per kilogram is determined, then obviously at 25 kilograms, we're already at greater than 17 log 10 inactivation. So gamma irradiation is very effective for coronaviruses in general, we would say. For rotavirus, again, we don't have published data on gamma irradiation of rotaviruses in serum, but we do have information on other reoviruses. That data is shown here. So for 25 kilograms, let me just stop and say the typical irradiation range for frozen serum is 25 kilograms to 45 kilograms. So 25 kilograms, we would expect to see 3.9 logs inactivation of a reovirus. And at 45 kilograms, we would expect to see 7.1 logs irradiation inactivation. So again, gamma irradiation is pretty effective for reoviruses. Bovine leukosis virus is a retrovirus, and we don't have much information for gamma irradiation of retroviruses in frozen serum. We do have information on human immunodeficiency virus, which is a retrovirus, in frozen human plasma. So it's a similar substrate, and it showed pretty good effectiveness. So we would expect, based on this data, that 25 kilograms would cause 3 logs inactivation, whereas 45 kilograms should cause 5.4 logs inactivation. It's pretty effective, not super effective, I would say. Schmallenberg virus is a Bunya virus endemic to cattle in Europe. We don't have data specifically for this virus, but we do have data for other Bunya viruses in serum. This is that house data from the USDA. Based on that data, 25 kilograms should cause 4 logs inactivation. 45 kilograms should cause 13 logs inactivation. So I would say gamma irradiation is very effective for Bunya viruses in general and should be effective for Schmallenberg virus. I'm not going to go through all this data. I wanted to put it in the slide deck. This is the data that we have for efficacy of gamma irradiation in frozen serum specifically, and it includes a variety of virus families, enveloped and non-enveloped. Here you can see. This is the more important table. It's looking at the same virus families, only this time we're portraying viruses of specific concern for a bovine raw material. And we're looking at efficacy at 25 kilograms, the lower end of the dose range, and 45 kilograms, which is the higher end of the dose range. And you can see for most of these viruses, we're getting significant inactivation all through this dose range. I want to point your attention to the bottom where we have parvoviruses and polyomaviruses are not very susceptible to gamma irradiation. You can see the problem there at 45 kilograms. We're expecting to get around 2.5 logs inactivation of the parvovirus and about 1.8 of the polyomavirus. Some important questions to consider. Can we rely on previously reported viral inactivation efficacy data for gamma irradiation? In other words, can we rely on that previously reported data to predict future performance of this barrier technology? So that really comes down to 2 questions. Do we have sufficient data to predict that similar inactivation efficacies will be displayed for all viruses within a given family? I would say the data are not comprehensive on this question, but on a mechanistic basis, it is not expected that we would see significant differences in efficacy among different members of a family. Secondly, do we have data for all virus families of concern? And I would say, no, we don't have complete data. For instance, we have limited data for retroviruses in serum. So there are some gaps. There's no denying that. And is there still virus risk in gamma irradiated serum? Yes, there is. As we know, bovine parvoviruses, bovine circovirus, and bovine polyomavirus, if in serum, is not likely to be totally inactivated by gamma irradiation at the kilogray doses typically used. Okay, you ask again, can we just use a higher gamma dose to kill all these viruses? Well, below, I made some predictions based on modeling to see how much, what dose of gamma irradiation would actually be required to inactivate 4 logs of these resistant virus families. So for parvoviruses, I predict 45 to 75 kilograys to inactivate 4 logs. For circoviruses, greater than 180 kilograys. And for polyomaviruses, greater than 100 kilograys to inactivate, again, 4 logs of these viruses. And what data do we have that we can't just go to higher gamma doses? Well, in 8 publications in which cell growth was monitored in irradiated serum, the maximum tested dose was 50 kilograys with a range of 32 to 50 kilograys. Serum performance at greater than 50 kilograys has not been characterized in the literature, to my knowledge. There have been anecdotal reports of higher affluences being used successfully, but I have never seen these in a publication. So for now, I would say that the maximum affluence consistent with maintaining serum performance is 45 kilograys. I'm going to wrap up now. Gamma irradiation will adequately mitigate the risk of most of the viruses of concern from the USDA, World Health Organization, and biologics manufacturing points of view. And I would include in that statement the more recent emerging viruses of concern. There are a few remaining viruses that will likely survive gamma irradiation, and these are the parvoviruses, circoviruses, and bovine polyomavirus. These are, some of these are common contaminants, but all of these have low likelihood of human pathology. I don't believe that these viruses are of great import concern, but I won't make that statement. Have I answered the question, who drives the validation bus? Well, the guidance is pretty clear on this. Serum suppliers making inactivation claims must validate the irradiation process and efficacy. Regulated users making claims as to barrier treatment efficacy must validate the radiation efficacy, and this is done in regulatory filings. Unregulated users, you just need to establish that irradiated serum will work as good as non-irradiated serum in your specific application. And the rest of these slides are the references that I spoke of before, some review articles, and I've given you here what these articles address. So for instance, efficacy, cell growth, efficacy, cell growth, or just efficacy. And this Hansen is mostly discussing regulatory drivers. Here are the serum supplier articles and what they address, efficacy, cell growth, or both. Some more on serum suppliers, regulated serum users. Here's the Raymond and Nims from Amgen, and here's the Razi vaccine, and regulatory articles. This is the 2 USDA articles and an article from the Paul Ehrlich Institute in Europe. And here's the bovine serum. So I hope that addresses the question that I was supposed to answer, and I will be happy to address any specific questions as they may come up in the next few minutes. Thank you very much.